Today we're going to talk about human organoids, one of the very fascinating emerging parts of science where neuroscience meets AI and potentially robotics. And one of these major discoveries are happening at the Indiana University. And I'm gonna read a couple of points that I wrote here for you, just to give you a little bit of a background before we start talking about brain organoids. So Indiana University began its work on integrating brain organoids with AI systems in December 2023, where they introduced Brainaware. It's a hybrid computing system combining electronic hardware with lab-grown human brain cells. I repeat, lab-grown human brain cells. Very exciting. This initiative is part of a broader field known as organoid intelligence, which aims to develop biological computing using 3D cultures of human brain cells. And there are other institutions such as University of Tokyo who are also in the space. They're exploring different types of integrations, focusing on constructing neural circuits tissues by connecting organoids through axons. Now, what Indiana University is doing is not totally new. In fact, they are piggybacking on existing research and existing body of knowledge, of course, such as stem cells. Nonetheless, the combination of what they're doing is pretty novel. And what they're working on for the organoids and AI integration in actuality builds on advancement in stem cells research, neuroscience, and bioengineering. They focus on creating Brainaware, which is a system that connects brain organoids to AI with MEA. And MEA stands for micro electrode arrays. And this is also not new, by the way, so it's been existing for a while, but nonetheless, the integration of these different disciplines is novel. The initiative started as a collaborative effort, I guess, across multiple disciplines, where they try to combine expertise in growing organoids from, again, as I mentioned, stem cells, with innovations in computational neuroscience. And we've already talked about computational neuroscience in this channel in the past. And the goal is to enhance the adaptability and efficiency of human brain-like neural networks to enhance AI systems, making them more energy efficient, adaptable, and capable of solving complex problems. And here's a breakdown as to how they are using these different disciplines to integrate and building this brain organoids. As I mentioned, one of the things that they use is MEAs, or micro electrode arrays, and they have been around. MEAs have been around for many decades. And their primary usage is to record electrical activities from biological tissues. And they are well established, for example, in neuroscience or biological sciences. And MEA allows researchers to capture real-time data from neurons or cells or organoids to see what their electrical activities is like. And these systems have been progressively refined throughout the years to increase their sensitivity and also their capability. These researchers also use brain organoids, which are 3D miniature models of the brain created from stem cells, and it's become much more exotic, I suppose, in my biomedical research as of lately. But again, it's nothing new per se. And they are being used to study the human brain development, different diseases, and also drug responses of the brain. While organoid technology itself has been significant advancement in recent years, it is built upon a foundational knowledge of stem cells, what we know of stem cells in biology and of tissue engineering. Now, how are these researchers combining brain organoids with artificial intelligence and neural networks? So the innovation is the fact that they are integrating the biological systems, in this case organoids and MEAs, with AI networks by recording the electrical activities of the brain organoids using MEAs. These scientists are generating data that can then be fed into AI systems. These AI systems can process the data, learn patterns, and potentially enhance their own learning capabilities. And these artificial intelligence neural network systems can also be used to interpret complex biological signals from the organoids, enabling better understanding of brain functions and disease mechanisms. Now, let's look at organoids' history. Where do they come from and how did they evolve? So the history of organoids is relatively recent per se, but their development has been a significant milestone in biomedical research. Organoids have evolved from foundational stem cell research and tissue engineering, and their uses have expanded dramatically onto various scientific fields. For example, in neuroscience, drug development, disease engineering, and disease modeling, and so forth. When it comes to the stem cells and early foundations, essentially in the 2000s, the idea of creating miniature and simplified versions of human organs stemmed and became very attractive to scientists in the fields, because it could potentially help the scientists to, in lab, work with these new build organoids to have a much more degree of freedom to conduct their research. Embryonic stem cells, for example, also stemming from stem cell research, were, for example, very pivotal in this area. These cells have the ability to differentiate into almost any type of cell in the body, which is amazing, laying the groundwork for creating organ-like structures in the lab, which is again amazing. And as I mentioned earlier, universities also in Tokyo have been very pivotal in this body of work. First major breakthrough in organoids technology came around in 2013, where scientists published a paper and they were able to grow intestinal organoids from human blueprint stem cells, which is again amazing. And they were able to provide the right growth factor and also made them suitable in 3D environments. They could develop complex tissues that resembled the organs that came from human being or different organs within the human brains, and they called them the mini organs. And that is how essentially it started. How are these organoids made, by the way? Organoids are generally made from stem cells that are cultured in a controlled environment within a lab, and the process of developing, for example, stems from deriving stem cells, differentiating these stem cells, 3D culture, and maturation. And there are very different applications in which we could use the organoids. Disease modeling is one of them. Pharmaceutical companies are increasingly using organoids to do the drug testing, as well as research, of course, and now most recently using them into connecting with AI technologies such as neural networks. Most recent development within organoids has been for example, regarding creating hybrid systems with AI. With hybrid system, we mean brain, organs, and AI neural networks. And there has been a growing interest in using organoids for research into creating these hybrid systems where biological systems are interfaced with artificial intelligence neural networks, as seen in research at places, as I mentioned, Indiana University, Tokyo University, and so forth. Another area is advanced in precision and complexity. Organoids research continues to become more sophisticated, and most recent breakthroughs include combining multiple organ types such as liver, heart, pancreas into one system to study how different organs interact with each other. Another area is gene editing. With advances in gene editing technologies, researchers can now create organoids with specific genetic modifications, allowing them to study rare diseases and model genetic mutations much more accurately. There are, of course, many ethical considerations that researchers need to be thinking about. Nonetheless, those ethical consideration is also being discussed a lot, and so we're not covering it in this video, but please know that if you feel like this is of a huge ethical issue, there is always ethical consideration when it comes to these type of cutting-edge technologies. Last year, I published a book called Symbiotic Intelligence: When Biological and Artificial Minds Merge. This work had been part of my work for the past 10 years, and the book ended up being over 600 pages, which is way too large or way too big. And I realized that is just a lot of people don't have patience to read big books. And so this year, my goal is to publish a condensed version of it, and maybe just making it half of the size at 300 pages. It is published on Amazon, and I have also included the link, as always, in the box below. But nonetheless, while I'm not particularly talking about organoids in the book, I do present a lot of not so far-fetched now ideas that for me started 10 years ago in terms of combining the biological intelligence and artificial intelligence through the electrical activities, through the wave-like electrical activities that we can harness via non-invasive technologies such as neuroheadsets and reading heartbeats and heart rate variabilities and so forth. And that is discussed extensively in the book. Alright guys, I hope you found this video useful. Thanks for watching. As always, like, share, and subscribe, and I'll see you next time. Bye.